Welcome to The Plunge, a cryo-EM podcast from Mimi, Mike, and Liz, sponsored by Thermo Fisher Scientific.
Hey guys!
Hey Melody! Welcome.
Thanks. So welcome to The Plunge. We're really excited to be here today with Melody Campbell from the Fred Hutch. She's going to tell us all about herself and her awesome work. I'm Mimi Ho and from Columbia University.
Liz Kellogg from Cornell.
And Mike Cianfrocco from University of Michigan.
So yeah, I guess without further ado, you started your lab during the pandemic. So I started in January 2020, you started your lab.
July 2020. Yeah,
Right,
a little
and
bit
so-
after you.
Yeah, and so I feel like both of us have this, or there's this whole cohort of junior PIs that started their labs in the middle of this kind of crisis where there's been like, lab supply shortages of people, like people not wanting to move or wanting to work from home, like all of this stuff. And I know it's added like an entire wrinkle to the experience of being a new PI. So can you talk a little bit of just kind of what was that like for you?
Yeah, I mean, for me, obviously, it was a really big adjustment for everyone to start adjusting to the ever-changing demands of the pandemic, but also I was adjusting to the demands of moving to a new city and trying to set up something brand new that I'd never done before. I think also for me, I had spent, the past whole year attending conferences, promoting my work, and then going through so many faculty interviews, traveling everywhere that I hadn't really been at the bench for quite a while. And so Yifan Chang was my postdoc mentor, and he's super, super supportive. So kind of the plan was from March until July, like that's when I was gonna start doing my preliminary experiments, start trying to figure out which equipment I needed, what I needed to order, like just little things like what brand tube rack is this and what brand, buffer is it that makes it so the experiments actually work. And so I actually wasn't able to do any of that because UCSF shut down so hard. So I think for me, that was definitely one of the biggest challenges. I didn't get that time to get any preliminary data, and all of a sudden I was in this big empty lab and I hadn't taken measurements of how big equipment is, and I didn't know what type of table to buy to put it on. So I think that was probably the biggest challenge.
Yeah, no, I remember for me, before I left my lab, I literally went around the lab taking photos of everything because I was
So
like, I'm
envious.
But I had the exact same thing where when I started, I was just I did not know that this was going to be my job, deciding between 5 different brands of 20 different kinds of up-and-door tubes, and I had this huge analysis paralysis thing going on that was kind of really difficult to get used to, for sure. What else, I guess, in terms of, did you have to deal with the lab supply shortages? Because for me, I got to stockpile stuff right before it happened, and so I had a ton.
Yeah, there were a lot of shortages for a lot of equipment was really slow to come in, obviously consumables, but actually I got kind of lucky where we had a relatively large and extremely well-stocked tissue culture room that hadn't been used for a couple years. So fortunately, I got things like filter pipettes and 15 mil conicals that I know that were really difficult. So in that respect, I think I actually got pretty lucky.
Yeah, no, that same thing happened for me. I got lucky that I did all my stockpiling before it actually hit, and so other people in the building were actually coming to me because I was like a hoarder. My entire lab was just cardboard boxes for a time. So yeah, I guess in terms of, aside from the lab supply issues, in terms of kind of setting up your lab and hiring people, what was that like in the pandemic for you?
I mean, I just feel like I always get so lucky. I like where my lab is. There's 2 other labs, Singvi Lab and BiLab, that are right in the same hallway. And those 2 PIs made a really big effort to make me feel welcome. Because otherwise, I think it could have been a really, really isolating experience. You don't really know anyone, but you're not allowed to come talk to anyone. So that was actually really, really nice where they said, just come into our labs if you need to look up supplies or if you need to borrow anything. Also, the Seattle MCB program, which is the program that I get my graduate students from, or one of the programs, they had a ton of really nice infrastructure that they set up really quickly to give grad students opportunities to meet PIs, meet each other. We still had lots of online symposium, so I managed to recruit actually.
That's great.
Pretty well too.
Wonderful.
Yeah.
Awesome. Yeah. No, I mean, because I know for us, when I started, we weren't taking rotation students in the lab because it was like all shut down, and so we couldn't take rotation students, right? And so that was actually quite tough because they were trying to do like a virtual one, and it didn't work very well.
Right, yeah, we have a lot of virtual rotation students, and I feel bad for those students because, they couldn't be around other people in the lab. They couldn't, learn organically. I feel like that whole, and even now they're trying to adjust to a lab full of people. Because they're so used to working alone. It's a very different experience.
Yeah.
Yeah.
So then I feel like, were there things that you felt like you did that really helped, aside from like obviously your immediate neighbors being really supportive? Like, was there anything that helped you with that?
For recruiting or just setting up the lab?
Both, and like dealing with all of the challenges of the pandemic. Did you feel like there were things that really helped you?
I mean, I think for me, making sure that I established a lab website and tried to make sure that, because people couldn't just come by and stop by my office, or I couldn't go, although the Zoom recruitment events were great, it's still it's always more intimidating to try to raise your hand on Zoom opposed to just stop by a poster or see someone grabbing a glass of wine or something. So I think for me, definitely making that lab website, saying who I am, what the lab philosophy is, what the research is, what our tools are, that helped a lot, I would say. I think it helped a lot for everything.
Well, obviously, I mean, you have a bunch of grad students in your lab, so clearly it worked. Whatever you did worked. So yeah, I feel like Mike was gonna ask you a little bit about.
Yeah, I guess before I
that
get
kind
there,
of leads into it.
I was actually going to just bring up something about sort of virtual recruiting. So that one thing at Michigan they did before the pandemic, they had this big kind of rotation student match-a-thon. It was like a big dating event kind of, where it was speed dating, changing tables. But it was all in one huge room. It was way too loud. And so it was kind of a problem. And now it's all on Zoom, and it's actually something that's better on Zoom.
Really?
Because it's they
interesting.
just swap rooms around for an hour, and you meet 10 or 15 grad students.
I want to do that.
And that was an innovation with Zoom. You're this is going to stay this way actually.
Oh, that's cool.
So it's interesting. There are things that change that are better. I mean, also remote work and working from home.
Yeah, absolutely.
I think it helped push all of our microscope usage too, to be we were kind of dragging our feet on remote access on the instruments. And I feel now it's you can do everything remotely. Which
That's definitely
also
true.
Innovations.
Yeah, no, ours were absolutely not. And then when the pandemic hit, they were well, I guess we have to let you. And now we've just never gone back.
Yeah, I know.
We just keep it that way.
I think also with this generation of students, and probably with us, for certain things, I'm just a lot more comfortable texting than definitely talking on the phone, but even sometimes being in person. And I think as a mentor, sometimes it's a lot easier for the grad student to have it in writing, because if you have an idea for a next experiment or something like that, I talk really fast, and then sometimes I see my grad students scribbling to write down what I'm saying, and then I try to say, okay, take your time, take a little bit slower. But especially at the beginning of the pandemic, I really just got into the habit of just sending them Slack messages all the time. And maybe it's kind of silly because my office is attached to the lab, so on the one hand I could go over there and bug them with whatever's on my mind. But on the other hand, then they have it in writing, so there's fewer possibilities of mistakes. And also, if they're in the middle of loading a gel, they don't have to deal with me being listen, I have this really great idea, and here's this paper, and I want you to read this, and let's do this tomorrow.
Yeah, no, I literally had the exact same experience where I noticed that my postdoc kind of whenever I come, she's in the middle of something, she's in the zone, and I come and I'm super excited, and I have these ideas, and they're just because I'm pulling them out of the moment, right? And so after a while with her, I noticed, then I also would watch her face and be oh no, I did something wrong. And so then it got really awkward. And so then I talked to her, I was do you want me to give you a heads up when I come into the lab? And she's that would be amazing. So now I Slack her and let her know that I'm coming whenever I'm coming to talk to her. And that's worked out really well.
Yeah. I guess it's all about communication, just good communication. In any case, so you start your lab, right? And
so I guess let's talk about your postdoc work on integrins. So what I remember is hearing you went to Yvonne's lab, and then hearing you're working on integrins, and I feel it was sort of I knew you were working on it and hadn't seen structures for a while. And then when I first saw you talk, I think it was at probably a Gordon conference. And it was just sort of all of these structures and all of the sort of things you discover on by themselves and bound to cofactors. So tell us about the structures. What got you through it, and what?
Sorry, one question. It was in Hong Kong, right? Was that the conference?
Yes, I gave one at the previous GRC too, the one on the East Coast.
Oh,
so there were
okay. I don't
actually
think I
two
was there.
separate integrin talks about two separate projects.
I remember just being blown away by all these structures and all the hurdles and all the things. And so tell us what that was like. I never really found out getting through that, what that was.
It was a challenging topic, definitely, just because integrins are relatively small proteins. They're about 150 kilodaltons. I was just working with a soluble construct, although they're supposed to be transmembrane proteins. So they're transmembrane, they're small, they're flexible, they're non-globular. They bind their ligands with really low affinity, so you have stoichiometry issues too. So basically, every single thing that's non-ideal for cryo-EM, integrins kind of embody. So we found a couple tricks to get around some of those issues, making it a soluble construct, picking the integrin that we knew to exhibit the least amount of flexibility for the initial experiments, and then studying it. Some of it was for a therapeutic standpoint by looking at it bound to Fabs first instead of the natural ligand, because those bind with a lot tighter affinity. So I think that the reason why I was drawn to that project was because it felt like the synthesis of the two semi-unrelated projects that I worked on during my PhD.
Since integrins are so flexible, are there any integrins that you've tried to study that are just recalcitrant to cryo-EM approaches?
So I think that,
or a single particle, I would say, current single particle pipelines.
I think that, we haven't been able to resolve all components of integrin, every single one of the domains. But what's nice is that it is a multi-domain protein, and there is one key beta propeller that we can usually at least resolve that. It also makes it so that can be like our fiducial for particle picking, or like making sure there aren't weird artifacts, because we always know we can at least get this beta propeller. Right. So that's not the most interesting bit of the integrin, but I feel like for that reason, it makes it so it's more incremental progress that we need to make. Like, we have a starting point. There's no integrin that we've tried to collect data for that we just can't see anything, that it's completely gone or completely destroyed or anything. So we certainly don't have structures for a lot of the integrins yet that we're interested in, but we just need to.
So good news, but a long way to go, right?
Yes, yeah. There's a lot to do.
So I guess one thing that Mike and I were talking about, like what kinds of things we wanted to ask people. And Mike happened to ask me, was there like a moment that really kind of like a pivotal moment or a pivotal person that really like made a big difference in your career or in your kind of life? And so for me, I was like, yeah, I do have a moment like that. And I think he was asking more about my PhD supervisor. And I was like, no, I didn't have anything like that with my PhD supervisor. It was actually happened right after I graduated from undergrad. So I was working as a research technician in Bob Stroud's lab at UCSF. And I had been there for about a year. And I had been working with this lab manager who had taught me basically everything, like all the basics of membrane protein biochemistry. And I thought things were going well. And then one day he sat me down and he was just like, how do you feel like things are going? And I was too young at that time to know that, like, that's a bad question. That's not a question you want, right? And so I was like, yeah, I feel like things are good. Like, I'm settling in, I'm learning things, I feel like I'm getting the hang of things. And he was like, well, I gotta be honest with you. We think you're a great person. We love having you around. I think you're really smart, you're really talented, but this is obviously not your first priority, right? And he was just like, and the thing is that, like, I think that you could turn it around, but if you don't do something different, if you don't start making this your first priority, I'm afraid there's not really a place for you in this lab, because we do high-level science here, and this needs to be your first priority, because what we're doing is important. And so for me, that was a huge moment in my life where I literally was like, okay, like I got myself together, like I started taking it really seriously. But I walked away from that conversation and I like had a lot of negative feelings towards him. I like really felt uncomfortable with him, and I avoided him for the rest of my time in the lab, which was like 3 or 4 years I spent avoiding this person. It was really obvious that I was uncomfortable with him. And then what happened is that, I moved on and I went through grad school. I went to Novartis, then I went to grad school, and then I eventually ended up in my lab in Columbia. And I was talking to someone looking back at it and being like, none of this would have happened if he hadn't taken the time to have that conversation with me. And I can see now from the experience, where I am now, that he did it because he cared, right? He did it not because he was being mean, but because he cared. And so I wrote this email to him where I was just like, thank you so much for doing this, for caring enough to risk me like not liking you. Basically this thankless thing that you cared enough to risk that and to set me right. And basically telling him that, all of this stuff would not have happened if you hadn't done that for me. And he then wrote back and he was completely he was just like, I'm so happy for you, that's amazing. He was like, there was like no bad feelings at all, right? And so I wanted to know whether you had a moment like that or a person like that in your life.
Yeah. So, I mean, I feel like all of my advisors have been really supportive over the years, and that's helped me get to where I am. But for a pivotal moment where it's kind of like I could go one way or go another way, I actually do have that exact moment. So I had just finished my undergraduate degree at University of Michigan. I had been an undergrad in Katrine Karbstein's lab for the past year. And I had looked at the statistics of being a PI, and they didn't seem very, favorable. And I also had a lot of self-doubt, and, professors seem like they're really put together and everything figured out, and they know what they want to do. So I had actually applied to pharmacy school. I thought this is, you go to pharmacy school and you basically become a pharmacist. So that seemed a lot safer. It was a lot less risky. I knew that I really enjoyed doing independent research. I liked making tiny little discoveries, but ultimately it just seemed like there were a lot of variables. So my advisor, Katrine, brought me into her lab and sat me down, and she closed the door. And even at that point, I knew this is not a good sign. I was trying to think of what the most expensive piece of equipment in the lab was and whether or not I had just broken it somehow. I thought it would be a size exclusion column. There were a lot more expensive things. But so she sat me down, and then she said, I know you've gotten into pharmacy school and you have your plans for next year, but I think it would be really great if you just stayed in the lab full time for another year. You can defer admission, you can finish up your project, and also the lab's moving to Florida, so you can come live in Florida and help us move. So as an undergrad.
Where in Florida, though?
In a city called Jupiter, Florida. It's close to West Palm Beach, kind of far from Disney World, but absolutely beautiful. Lots of sea turtles. Cool. So yeah, as an undergrad, I wasn't completely set on what I wanted to do anyway, so that sounded like a really good plan. Yeah. And so I think, she knew me enough to know that if she could trap me for a year, that I wasn't going to go back to pharmacy school. So that was probably one of those moments where it really could have gone either way. And after that, I just kind of took everything one step at a time.
Have you talked to her since then? Have you ever touched base again?
Yeah, I have. And she knows that story. She knows how appreciative I am of that. Yeah.
That's awesome.
Yeah.
So I guess in that vein, what's next for you? So you have you've set up your lab and everything. I guess I actually wanted to ask you, if we have time, what made you fall in love with cryo-EM? Because I feel like we all have that one moment. For me, it was the first time. I was so used to looking at protein on a gel filtration column, and then it's abstract. It's because there's a smooth peak, that means your protein is well behaved. And so it was that moment when I first did negative stain. I put it into the F20, and I was like, oh, I can visually see, it's aggregated, or it's well behaved, right? That was the moment for me. So what was the moment for you?
Very similar, actually. And it's good that I already told that story about Katrine and how great she is, because this part of the story, so I'm a biochemist, she is a biochemist, I was a biochemist in her lab, but she had a collaboration with Yorgos Skiniotis. So I would hand protein off to the Skiniotis lab, and the first time I did it, the next day popped up in my email an image of a micrograph of that protein that I purified the day prior. So although obviously I'm still a biochemist, I was just I want to do that part. Yeah. That's what I want to be doing. So then when I applied to graduate school, I was actually very focused, where I wanted to be in a cryo-EM lab. I wanted to do exactly that.
Yeah, no, I feel the same way about visually seeing things. And for me, it was when I started grad school and saw it myself. But I remember textbooks had negative stain images and some cryo images of ATP synthase or different things. I remember just being that's what proteins look like. Because before then you look at PDB models and crystal structures, and I felt abstract, and there's something about seeing an image of a protein that was
That's so funny you say that, because I really like the aggregates. Because I was interested in, I was studying protein folding, and I was always interested in what happens when they misfold. And so for the aggregates, I was that's what a misfolded protein looks like.
Right.
And I was is there a way we can understand it?
That's
maybe
true.
for the way future, but I was pretty fascinated. I had no idea what a misfolded protein looked like.
Right. Well, I think it sounds kind of cliche, but ultimately a picture is worth 1,000 words. And that's very much it.
Yeah, especially the aggregates. I feel you look at the aggregates and you're oh, that's my protein all piled up. It's not some amorphous thing that it's all right there and I can't use it.
Yeah. I was there's information here. here that we're not, you can't get in any other way. I thought it was really amazing.
That's so funny. Like the rest of us are like, oh, we screwed up our protein prep. We have to go do it again. And you're like, this is so cool.
You're like, can I take
I'm gonna save that image.
Are you like gonna take a tomograph of that next? Is that what you're thinking? You're gonna take it and like do?
I don't know. I don't know if we're there yet, but sometimes I wonder,
I just love how we have a similar thing where conceptually at the time crystallography seemed like one step too far removed. Like
Yes.
diffraction patterns, how do we go from crystals to cryo-EM? It seemed so much more straightforward in cryo-EM, image to 3D model, but then when you actually get into the methods, you realize it's just as much of a black box when you're starting.
Yeah, it's like step 1, Fourier transform, step 2, command line, and you're just like, whoa, and it starts flying at you.
First thing you do is take it out of real space. Yeah.
But as like a naive, brand new student, you're like, I can actually see everything that I'm doing. It's like, no,
I mean, circling back, that was also probably one thing that was good for recruiting. I feel like we have it easier than some other fields where it's easy to convince students with these beautiful high resolution structures and be like, you want to know what the vaccine made and you want to know what your body is making against a SARS-CoV-2 spike. Like, look, here it is. You can see exactly how each variant is changing and how your body's making better antibodies.
I think one example of that stands out at the Gorn conference last fall, Peizhu Zhang showed this like a cell that was what they administered the AstraZeneca vaccine. On the cell membrane were all these spike proteins lined up in like a hedgerow, and you're like, whoa. It was
It's
like
happening in your
one
body.
of the coolest things from that meeting. I was like, that's
Yeah,
incredible.
because you hear that that happens, but then when you see it, you're like,
You're like,
ugh, my
whoa.
cells are
Exactly.
That's absolutely true. So I guess kind of just to wrap up, what's next? Like what are you super excited about right now?
Well, I mean, probably everyone that you're talking to is giving the same answer, but tomography, cryo-ET, that's the next step, I think, for everyone. I mean, it's really important to look at these proteins, get high resolution structures, but if we can link that with cryo-clem, knowing where things are in the cell, and then fitting higher resolution, maybe single particle structures into lower resolution cryo-ET, we get everything in its native context. We don't miss out on any critical binding partners or anomalies that you might see when you just have protein by itself in a buffer. So my lab, as well as probably everyone else's, is starting to look into how do we incorporate that into our own research to really just give the complete picture. Because I feel like right now with all the advances in light microscopy as well, you can get up to a certain point, but then there still is that little gap. And then you have single particle and crystallography. But if you just fill that gap, you get the continuum.
Yeah. No,
And so you sort of made the case at the beginning that integrins are so small and hard for single particle. So like, how are you thinking for tomography?
Yeah, so I mean, they're expressed on the surface of the cell, so looking at how cells interact with each other, how viruses interact with integrins on the surface of the cell. I think right now with our current tools, we're probably not going to be getting really high resolution structures of integrin in their native context. We'll get the lower resolution envelopes, and then we'll be able to fit our higher resolution models in there, or be able to track exactly what state the integrin is, and we still need the single particle data to make those conclusions.
As part of this series, we're actually interviewing Bridget later in the day, and I know that you did your PhD work with Bridget and Clanet Scripps. So, and it's also an interesting environment, right? Because, but you can tell us more about it. What was it like doing a PhD with Bridget and Clanet Scripps?
Yeah, so I think that the lab actually is a little bit atypical in that traditionally they have about 2 grad students and 2 postdocs at any given time. And then on top of that, we have senior technicians, we have engineers, software programmers, things like that, where it's kind of not exactly the biochemistry, biology driven lab that many other people go through. But I think for me that was a really exciting environment because I wasn't really, all I knew is that I wanted to do cryo-EM. I'd worked on ribosomes before, so I thought I wanted to work on ribosomes. But what was really cool is that because that's a collaborative lab, I actually got to work with many different groups. So I started out working on technology-driven experiments, but then by the end I was working on the nitric oxide signaling pathway. And that's actually what led me to become really interested in these super flexible heterogeneous proteins.
If I can jump in then. So, you said technology developments. I remember when your Eli paper came out on the proteasome, that was a landmark paper because of the resolution. Revolution was still kind of happening. So do you want to tell us what it was like approaching that and what it was like to see structures at that resolution?
Yeah, it was really, really exciting, and it was very much a group effort. So I think at that point in time, it was really exciting to be in the technology field because we had that new direct detector. So now we could get movies and we could get higher resolution that we ever really thought was possible, and we could collect more data, and the software was being improved so much that on the one hand, it really felt like a race. But on the other hand, we were all helping each other out so much. So when I started doing that technology development, I was working with Nico Grigoriev. For the proteasome paper, that proteasome actually came from Yifan Cheng's lab. Like, although we could have been viewed as competitors, he was extremely generous. And as soon as we asked, he said, sure, here's a great biological sample that you can use. And actually, for that structure, we collected a lot of data sets. It was still bleeding edge of understanding that detector. So we had a lot of data sets that did not go as well. But ultimately, it was due to the co-first author on that, David Wiesler, where he was trained as a crystallographer. And as we were modeling it, he was like, oh, those are water molecules.
Oh, wow.
So that was a really, really big moment that I think, as someone who's only been trained in cryo-EM, I might have just hide dust, like we don't even see that.
Exactly.
So it was really this big collaboration of the cryo-EM field as well as bringing in crystallographers to really, use some of the existing tools, use all of the existing tools to make that structure possible.
Yeah, definitely. Do you feel like part of the reason that happened was because you were in Bridget and Clint's lab, and they have part of the reason it was able to be successfully so collaborative is because they had all of these. They already kind of had that going.
Absolutely, yeah. They had the knowledge of the optics and the software and the data collection. With Anqi Cheng there, she could just write that software on the fly that could accommodate the movies and the bigger files and all. I mean, that's one thing to mention, is that before we were just using these small 4K by 4K single CCD detectors. Yeah. Now all of a sudden you're taking 50 frames, so each image is 50 times larger. Yeah. So it was really both technology and methods and biology and know-how, so it was really a group effort.
It must be really fun to be like a graduate student in that environment where you have all of these experts and you have access to so many experts.
Absolutely, yeah, and especially exciting because everything was so new, so even if it didn't really work, it's still a result, right? Yeah. People are still interested, and we tried this dose weighting that didn't work, or we tried these box sizes and it didn't make a difference, where it was just exciting that it felt like everything was very productive at that time.
And I think to go off of sort of the community going into that structure, what was nice is that you guys turned around and then released your data set onto sort of the emerging Empire database, and you're, one of the few first entries on there that everybody is now analyzed through different tutorials like CryoSpark. So I think that's also a testament to your work and to the group there.
Yeah, there's definitely a moment though when the first structure came out using that data set that was much better resolved of the ones that we had, but obviously the software over time improved. Where now I think pretty routinely from a pretty small subset, you can beat our 2.8 angstrom that we were very proud of at the time.
Well, I always have all my students analyze that data set to start with.
Oh, fantastic, yeah.
It was great talking to you. Thanks for coming out. It's been really nice hearing all your work.
Yeah, thank you so much for the opportunity, and hopefully we'll get to chat more later.
Thanks for listening to The Plunge, a cryo-EM podcast from Mimi, Mike and Liz, sponsored by Thermo Fisher Scientific.